Assalamu alaikum. Today I will deliver you lecture on the topic hepatitis C virus. The last lecture I ended my lecture due to childish activities in the Zoom cloud. Anyway, today lecture is on the topic hepatitis C virus. This virus previously was known as non-A, non-B. Because the some of the cases were those cases in which the blood was donated or the transfusion were made. These cases were infected with the virus, which was neither A nor B. So this virus was named as non-A, non-B. However, in the current days, nowadays, it is called hepatitis C virus. This virus belongs to a family that is known as Flaviviride. And the other viruses which belong to this family, these are the yellow fever virus and the dengue virus. So yellow fever virus, the dengue virus and the hepatitis C virus, they belong to this Flavi family. And this virus, hepatitis C virus, is that this also belongs, this was the family and this is the genus Hepatitis C virus. So this is also known as the Hepatitis C virus. Hepatitis C virus. This is a singularly stranded RNA virus. You will be aware that the hepatitis B virus was DNA virus. That was the DNA enveloped virus, while the C virus is singularly stranded RNA enveloped virus, while the hepatitis B virus was a partially double stranded DNA virus. This is also known as the non-A, non-B virus, and unlike to the hepatitis B virus, this C virus has no polymerase enzyme, while the B virus has the polymerase enzyme. So, regarding the structure of hepatitis C virus, they are the different antigens and the proteins, and these are divided into the structural proteins and non-structural proteins. The structural proteins, these are the core protein known as the C antigen, and the envelope glycoproteins E antigen. So these are the two antigens or the proteins. Core protein is a viral capsid protein, and E for envelope. So E determines envelope, C determines the capsid. are the core. And the non-structural proteins, NS means non-structural protein. These are divided in different numerous proteins, NS2, NS3, NS4, and these are the different antigens. This is the structural outline, the antigen, enveloped antigen, E1, and the capsid antigen, C protein, and the genome, nucleic acid genome inside the envelope and inside the capsid. There are various genotypes. They have the importance, they have clinical importance because the treatment varies according to type, genotype. And genotypes are determined, they are based on the RNA sequence analysis. The sequence of the RNA, the genome of the hepatitis C virus, the nucleotide sequences. On the basis of nucleotide sequences, they are divided into various genotypes, and these genotypes are also called the clades. So the other name for the genotypes is the clades. Clades differ from each other by 25 to 35% at the level nucleotide level. So there are the 11 genotypes. Worldwide, there are the 11 genotypes.
Hello.
From G1 to 11.
Hello.
Worldwide.
Sir, hello.
Hello.
Hello. Sir. सर हमें लाइसेंस मिल गया है तो सर आप जो है वो अभी भी 100 पार्टिसिपेंट्स वाला ही जो है यूज कर रहे हैं, प्लीज आप लाइसेंस वाली लिंक यूज करें ताकि हमारी पूरी बैच इन्वॉल्व हो सके।
क्या में आईडी?
सर जो आईडी जिस लिंक पे आपने यूज की है वो पुरानी वाली है, जो बगैर लाइसेंस वाली है, इस पे सिर्फ 100 पार्टिसिपेंट्स इन्वॉल्व हैं।
तो मैं आपके वो आसानी के लिए वही आईडी यूज कर रहा हूँ, अगर आप कहेंगे तो मैं तो चेंज कर दूंगा, कोई मसला नहीं। मैं उसलिए कि पहली मर्तबा मैंने वही आईडी रखी। बेटे वो उसी आईडी पे
सर इस लिंक पे इशू ये है कि इस पे सिर्फ
हाँ जी बेटे सुनाओ।
सर यहाँ पे सिर्फ 100 पार्टिसिपेंट्स जो है वो सर यहाँ पे सिर्फ 100 पार्टिसिपेंट्स ही आ सकते हैं, इससे ज्यादा जो सकते हैं।
अच्छा तो 100 पार्टिसिपेंट्स आते हैं, वो तो उससे ज्यादा उन्होंने।
प्लीज एक दफा आईटी वालों से कंफर्म कर ले, ये वो लिंक नहीं है जो आप यूज कर रहे हैं इस पे।
अच्छा देखो, तो ये लेक्चर तो मैं डिलीवर कर दूं ना बेटे, क्योंकि आईटी से तो फिर टाइम चला जाएगा। अभी आईटी वाले तो अपने डिपार्टमेंट में है, मैं अपने घर पे हूँ बेटे। वो तो आईटी वालों को फिर बताऊंगा कि मैं हर दफा नई आईडी रखूं, क्या करूं, क्योंकि उन्होंने तो जो है 100 से ज्यादा स्टूडेंट वाले मेरे ख्याल में वो तो ऐप उन्होंने अभी, मुझे पता नहीं है कि उन्होंने लिए कि नहीं लिए।
सर ले लिए, हमारे सारे लेक्चर्स वहीं पे ही होते हैं, सिर्फ ये आपका लेक्चर यहाँ पे हो रहा है।
अच्छा अच्छा बेटे ये लेक्चर जो है फिर मैं जो है ना पोस्ट कर दूंगा यूट्यूब पे बेटे, कोई मसला तो नहीं होगा।
अच्छा
मैं यूट्यूब पे पोस्ट कर
ठीक
दूंगा
है,
बेटे। इधर फिर मैं वो देखता हूँ, वो जो नई हर दफा जो नई आईडी मिलती है मैं उसे पे करूँगा। ठीक है बेटा?
ओके सर।
हाँ ठीक है।
ओके,
बाकी ये
थैंक्स सर।
यूट्यूब पे मैं आपको भेज दूँगा।
ओके सर।
So anyway, there are the 11 genotypes worldwide. 11 genotypes from G1 to G11, and the most common, the major are the G1 to G6. Again, these genotypes, they are divided into the subtypes, and there are the more than 100 subgenotypes. The genotype 1 causes 75% of the infections in the USA. So this is the common genotype in the USA. This is the distribution, geographical distribution of the genotypes. The genotype 1, 2, and 3, they are worldwide, and G1 accounts 40 to 80% of all cases of the world. And this is the geographical distribution of genotypes. Which genotypes is common in which country and in which area. How this hepatitis C virus is transmitted to the human beings. So here is the acronym BBS. You are an MBBS, but this is a BBS. Transmission is by 3 ways, and for memorizing the transmission mode of transmission, the BBS means B for blood, and the next B for birth, and S for secure. So these are the 3 modes of transmission. It is transmitted through primarily through the blood, in the blood products, in the blood transfusions. The most cases, 95% of the post-transfusion hepatitis are caused by hepatitis C virus. So the post-transfusion hepatitis, most of the cases of the hepatitis which are caused by the post-transfusion, exactly or more than 95%, they are caused by hepatitis C virus. And through the intravenous drug abuse, the parenterally, and the birth means the vertical transmission from mothers to the babies during the perinatal period. This is common. This is the common mode of transmission in the Asia, while the others modes of transmission, they are common to the other countries. So the third one is the sexual mode of transmission, and there is no transmission via placenta or the breast milk, and this is unlike to the hepatitis B. Hepatitis B virus is transmitted through the placenta or the breast milk, but here is just opposite. How the infection causes the disease. These are the type 4 immunity is involved, in which the CD4 and CD8 cells are involved. So you know, in case of the type 4 immunity, these T cell mediated immunity is involved. However, there is no cytopathic effect. Cytopathic effect is common in the acute hepatitis, while in case of the chronic hepatitis, there doesn't occur the cytopathic effect. So what happens in the infection of hepatitis C virus when this leads to the chronic hepatitis? There is a persistent viremia, then chronic inflammation, and this leads to the necrosis and fibrosis, leading to the cirrhosis, and then the hepatic carcinoma. So remember that hepatitis C, is the virus. C for the chronic hepatitis, C for cirrhosis, C for carcinoma. So this is the hepatitis. This is the virus which causes the chronic infection, and then the cirrhosis, and then the hepatic carcinoma. So the letter C is for determines the three conditions. You will be aware that the hepatitis B virus, the vaccine is available, while in this case, in case of hepatitis C virus, there is no, there is no availability of the vaccine. Why this is so? This is because of the genomic diversity. Genomic diversity, the genetic variability is responsible for unavailability of the vaccine. So this virus has the variability, at the hypervariable region in the envelope protein, and so this changes its antigenic shape, and that is why still the scientists, the microbiologists, they are not uh, able to discover the vaccine. And so there are the different genotypes in the different parts of the world. So these are the causes as to we are unable to discover the vaccine. Causes species, these are the subspecies. When the virus undergoes the sequence variation during the chronic hepatitis, this complex viral population in the host is called causes species. So these are the subspecies and known as the causes species in the host.